Feeling good, wanna feel great. Longevity starts, don't hesitate. Too much noise, what do you choose? Trendy hacks, conflicting news. Skip the myths, come hear what's true. From world-class experts straight to you. First and third Thursdays, 7 p.m. Science, health, and our team. Brain to heart, from food to rest. Live the talk on living your best. At 7, we'll see you there.
In NUS, have used the same sleep tracking device on 3 groups: undergraduate freshmen, working staff in NUS, and elders living in the community. When you look at the activity sleep plots, you think the ages are flipped because the seniors that participate in these studies sleep better, they are physically more active, especially in the morning, and they're less sedentary. I think this should raise alarms in the public health sector. Really, it's something that we need to work on.
The pressure that gets put on kids when they're early in life to perform well, and the effects it has on their sleep patterns.
Sleep is important for optimal cognitive function, and it's also very important for mood. Mood and motivation are something that affects learning, it affects social interactions, it affects our effectiveness as a unit in a community. So it's important to pay attention to sleep.
Welcome to the National University of Singapore Yong Loo Lin School of Medicine Healthy Longevity webinar. Thank you for being with us. You just saw a short summary of our last episode with Professor Michael Chi about the powerful geo-intervention, and that's called sleep. You just heard sleep is interfering with everything. So hopefully after this episode, you will have good sleep. The full episode is of course available on our YouTube channel. Today, we are talking about another geo-intervention, and that's the drug Rapamycin and the immune system. Assistant Professor Matt Yousefzadeh is joining us. Please send us all your questions, remarks, comments, whatever you want. Kalisha Yusri is going to moderate the Q&A session, and she will introduce herself first.
Oh, hey there.
Oh, hi. I didn't see you there. Come in.
Thank you so much. Can we know more a bit about you?
Yeah, so I'm Kalisha. I'm a research assistant, and I just became a PhD student here at the Healthy Longevity Translational Research Program at the National University of Singapore. And my PI is Vincenzo Sorrentino.
Congratulations on becoming our newest PhD student.
Thank you.
So what's your research about?
So I focus on a disease called non-alcoholic fatty liver disease, which affects around 30% of Singaporeans with a prevalence in the elderly. And of course, with an increase in these unhealthy calorie-rich diets and also with an aging population, we expect this number to go up even further. Currently, there's only one FDA-approved drug against fatty liver disease, that is resmetirom. So we're looking at other therapeutics that could combat this disease. The one I'm focusing on is trigonelline. It's a natural compound that exists in the human body and also in some plants like fenugreek seeds and coffee beans. Trigonelline is a NAD precursor, and you might have heard of other NAD precursors such as nicotinamide mononucleotide, NMN, or nicotinamide riboside, NR. So what is NAD? NAD is an essential cofactor that is involved in many cellular processes such as respiration, and it also tends to go down during aging in some organs such as the liver. Hence, we want to look at the effects of trigonelline in fatty liver disease. So when we supplement with trigonelline, the NAD levels in the liver go up, and this might ameliorate the disease. And you can see some of my results over here. So these are 2D liver cells that I have induced fatty liver with by adding some fats and oils to the cells. They're stained with 2 dyes, a blue dye to stain the nuclei and a green dye to stain the fat droplets. So on the left is the fatty liver, where the green dye is really quite abundant and high and quite intense. But on the right is fatty liver cells that are also treated with trigonelline. So you can already see that the number of fat droplets have decreased and the intensity of the stain is also decreased. And these results make us quite hopeful that fatty liver might be ameliorated by trigonelline treatment.
Thank you so much for sharing that. So what's your usual work life is like in the lab?
So along with modeling fatty liver disease in 2D cells, we also model fatty liver disease in mice by giving them a high-fat diet that is also supplemented with trigonelline. So by the end of this mouse experiment, we'll see if trigonelline is able to ameliorate the disease in a fatty liver mouse model. Along with that, I have also recently taken up 2 final year project students. They're year 4 undergraduate students, and so my day involves a lot of teaching.
That's really lovely. What's your biggest takeaway since joining our HLTRP program?
My biggest takeaway is that research is never done alone nor in a vacuum. I've met and collaborated with so many amazing scientists. For example, Professor Henry Yu, he has a model where you literally put an organ on a chip, they call it. So he models the liver on a chip, which makes it a lot more physiologically relevant and gives us some really cool results. Also, we collaborate with Dr. Caroline Wee, where she models fatty liver in a zebrafish embryo, which is also super cool and also gives us another animal model to validate our results in.
That's so great. And outside of lab work, what do you like to do during your free time?
I love to travel. I've recently picked up a new hobby that is scuba diving, so I scuba dive wherever I travel. Other than that, I love to keep fit in the gym, and I also love drawing, so all of the images that you see in this video are drawn by me.
You're so cool, Kalisha. Thank you so much. One last question. How can we follow you for more?
So you can add me on LinkedIn at this link, and also later I'll be moderating the Q&A session, so if you have any questions or comments, please add them in the Q&A box.
Thank you. See you.
Bye.
Thank you so much, Kalisha. I love the trigonelline study and, of course, the story and the scuba diving. But now first going to our guest today. Today's guest is Assistant Professor Matt Yousefzadeh from the Department of Medicine at Columbia University Irving Medical Center. And he has a background, of course, in aging biology. He studies how senescent immune cells affect the aging process. And his group focuses not only on animal models, but also on human samples to establish biomarkers of aging and to test potential gerotherapeutic interventions. I think after the fantastic RDD in Copenhagen, I cannot say gerotherapeutic anymore. But Matthew, it's great to have you here in the show. We are really looking forward to talk about rapamycin. I have lots of questions for you already without having seen your presentation. The floor is yours.
Thank you so much for having me. I'm really excited to be here. Let me pull up my slides. Yes, I'm in the Birch Lodge Center for Human Longevity here at Columbia. And our lab is really interested in immune senescence and immune rejuvenation. And just to quickly start off with, we need to give credit to the people that do the work. And so these are the individuals in my lab or associated with my lab that have done the work. Frederick, who's done a lot of the bioinformatics, Ritu and Alina, who have done a lot of the flow workup, and Tom, who has done some of the human clinical work that I will show you at the end. But overall, why are we interested in studying human aging, and especially aging within the immune system? And it's really important because the immune system is circulating throughout our body, so it's able to sense changes that occur throughout the organs in the periphery. As we get older, we start to lose our effectiveness of vaccinations, as well as we increase our risk of infections. And so this is really a reason to study the immune system. Another reason, too, is that a lot of groups are using epigenetic clocks as a surrogate biomarker for biological aging. Well, these are blood-based markers that are, by and large, driven by immune cells. And so when we look at this process of this degenerative decline in immune function, which is known as immunosenescence. And so you probably, the viewers have seen this hallmarks of aging before. And one of them is cellular senescence. Well, there's one for immunosenescence, which is again, that progressive related decline in immune function. And immune senescence, of course, falls underneath that umbrella, but there's a lot of different changes that occur too. Mitochondrial changes, cell population changes, where we start to lose lymphocytes and we have a myeloid skewing, loss of naive T cells, a buildup of memory cells. You'll also get lymphoidal atrophy, as well as this general increase in inflammation that can drive inflammaging. And so with immune aging, or immunosenescence, what we can see is that as we get older, our immune system starts to have a loss of immune surveillance. And immune surveillance is really important because in the context of aging, our immune system is really responsible for finding these pathogenic cells of aging and clearing them out, including senescent cells. And this can be done by a number of different cell populations, whether it's T cells, B cells, macrophages, and so on. And when you lose this capability, you get this accumulation of senescent cells that can drive inflammation and aging, as well as this loss of immune surveillance is what can allow for neoplasms and cancers to sort of sprout and cause a pro-growth environment for them. Also, we have a loss in Tregs, and Tregs are regulatory T cells, which have an immune suppressive function, which is really important for the resolution of infections, as well as they're necessary for preventing autoinflammatory conditions by driving tolerance. And so with aging, we can also see a loss of this Treg function. Similarly, in immunosenescence, immune cells can adopt pathogenic features. So T cells and B cells can become pro-inflammatory, as can innate cells like macrophages. Again, this can drive inflammaging, as well as we start to see a decrease in T cell and B cell receptor signaling. And these immune cells, these adaptive immune cells can start to behave sort of like innate immune cells and again become hyperinflammatory and drive tissue inflammation, which can prevent tissue regeneration. And another feature that's kind of been appreciated over the last 5 to 6 years is that premature immune aging can drive systemic aging. And so just to show you a little bit of this, when I was a postdoc, we worked on mouse models of immunosenescence, and we found that endogenous DNA damage, which occurs naturally, but you can enhance for by getting rid of a DNA repair enzyme, in this case, ERCC1, in hematopoietic cells, which are shown here in red are the cells that lack DNA repair. In the immune cells, these mice start out fairly normal at about 3 months of age. But over time, they develop progressive lymphopenia. So they're losing lymphocytes. And this appears to be largely B and T cells. There's no change in macrophages or granulocytes, as well as we start to see at about 5 months of age, there's a decline in immune cell function. And so this could be things like delayed type hypersensitivity, where you challenge the mice with an antigen and they don't respond very well, as well as the production of antigen-specific antibodies. So this could be used as a surrogate for vaccination. And lastly, we even see reductions in NK cell cytotoxicity. And this is really important because we know that NK cells are quite important for the removal of senescent cells. And so again, these mice develop a really robust immunosenescence phenotype. They express high levels of P16 and P21. Of course, they show DNA damage, inflammation. We see lymphoidal atrophy. So what we end up with is this really nice mouse model of immunosenescence. But going forward, we were really interested in the non-lymphoid organs. So we know that at about 5 months of age, there's premature aging in the immune system, but what happens in the rest of the mouse? And so if you look at the other tissues in the mouse, what we can see compared to the control mice, which are in this blue dashed line, the mice that lack DNA repair in their immune cells, they start to build up a high level of P16, which is a cellular senescence marker, a really classic one that's used to track cellular senescence, throughout their tissues And what we see is this isn't just senescent immune cells, which can be resident in those tissues. We actually found parenchymal senescence by staining for tissue-specific markers and looking for co-localization of P16. So if you take, for instance, the liver, we can stain for albumin, which stains hepatocytes, and we can see P16 staining in there, showing that it is true hepatocyte senescence, and we've done this validation in other tissues. And this starts to occur at 8 to 11 months in these mice, so well after the onset of immunosenescence. And when you look in some other tissues, let's say the liver, the kidney, intervertebral disc, or the muscle, we see these cell non-autonomous effects. So in the liver, we can see blood-based markers of liver damage, the aminotransferases. The kidneys, we see increased proteinuria, which is indicative of kidney dysfunction. The intervertebral disc, we can actually see senescence in them themselves, and we see impaired muscle regeneration after wounding there. And so what we think is occurring is a mechanism for the spread of immunosenescence to driving senescence in the periphery is that you have healthy immune cells, and they can become stressed either through DNA damage, or Maria Middlebrun's group has shown in T cells specifically, if mitochondrial dysfunction occurs, it can drive a very similar phenotype. That these senescent cells produce a lot of SASP or inflammation that can cause the normal cells in the tissue to become senescent, and this can drive premature aging. At the same time, we see this loss of immune surveillance, and this again sort of further reinforces it, which can ultimately shorten health span and lifespan. And so what we sort of say is this part of the the model is that we're building up a lot of trash in the body. But because the immune cells are becoming senescent and dysfunctional themselves, they're losing their capability to take out the trash. And overall, this is causing quite detrimental effects. But there is hope. Can we rejuvenate an older immune system? And the answer is yes. There's a lot of different ways to do this, whether you're using therapeutic antibodies for immune checkpoint inhibitor blockade, small molecules, CAR T cells that may target pathogenic cells of aging. And one of the ones I want to focus on is inhibition of mTOR with rapamycin. And so rapamycin is very interesting. It's where I work at, I'm in a bone marrow transplant unit, so it's typically used in very high doses for immunosuppression to prevent graft rejection. However, at low doses, it can be not only immune stimulatory, but other groups showed about 16 years ago that rapamycin can actually extend lifespan in mice. And this can be done with late life interventions, early interventions, and it's one of the most reproducible gerotherapeutics. Well, around the same time that this study was shown, another group, Chongqing and Pang Zin, had shown that rapamycin treatment before challenge with the H1N1 influenza vaccination significantly increased survival and rejuvenated older hematopoietic stem cells, or HSCs. And moving forward into humans, right as I was starting my postdoc and really interested in all of this, Joan Manick had shown in older individuals that were given a short course of a rapalog or an analog of rapamycin before they were given the annual influenza vaccination, it was able to increase titer levels against the influenza by 20%, which is incredible in older individuals because they typically are much poorer responders to vaccinations. And in fact, we have to give higher doses of vaccines in older individuals that we would give to younger people. And furthermore, they found that rapamycin treatment reduced expression of PD-1 on both CD4 and CD8 T cells. And PD-1 is a marker of immune exhaustion. And so for us, I wanted to test this out in our immunosenescent mouse, and where we looked at immune function. And so in blue are the control mice, and in red are the immunosenescent mice. And one thing that you can do is sort of like a tuberculosis test. You can sensitize mice to an antigen, in this case, keyhole limpet hemocyanin, and then you test them later on with a foot pad challenge, and you can measure foot pad swelling. And this is delay type hypersensitivity, which is a T cell response. And so you can see when challenged with The control mice, when challenged, they have foot pad swelling. However, the immunosenescent mice don't. This deficit is partially restored with rapamycin treatment. And similarly, if you look for antigen-specific antibodies, we're able to partially rescue some of that with short-course rapamycin treatment. We're able to reduce markers of P16 in peripheral blood cells, as well as reduce some of the SASP or inflammatory factors like MCP1 and TNF-alpha, which isn't shown here. So there's a lot of disparate data out there on what immune cell types are rejuvenated by mTOR inhibition. We saw some data from human T cells. Some other work has been done with gamma delta T cells, but we were really interested in taking an unbiased approach. And we were really fortunate to be able to get samples from the Rejuvenome project. And the Rejuvenome project was to take mice later on in life, it's say 18 months, and put them on rapamycin, collect tissues and do multi-omics analysis on them to understand better what rapamycin is doing throughout the body. Unfortunately, that project was terminated early, but we were able to obtain samples, spleen samples, from 25-month-old mice that were either on control chow or chow that had rapamycin, as well as we had young mice, and we had both male and female mice so that we could start to investigate sexual dimorphism, or sex as a biological variable. And so what we were hoping to look for is young versus old effects, the effect of RAPA, and really to understand, does RAPA restore a more youthful expression pattern, in particular immune cell types. And so we did this by doing single nuclei RNA-seq from these cryopreserved spleens. And so what we quickly found out is that spleens produce a lot of RNases, very likely because of neutrophils. And there's a lot of work out there showing that people will deplete neutrophils from cells before they actually do RNA-seq. So we had to sort of quadruple the amount of RNase inhibitor that we added in. And what we get is a number of different immune cell types that were captured in our analysis. And just showing you here the sort of different breakdown of these groups, and the young mice, the old control mice, or the old mice that were treated with rapamycin. And so there, looking at this volcano plot, we can see effects in B cells that are either genes that have increased expression with rapamycin treatment or less. And so with less, it looks like Ki67, which is a marker of proliferation. With rapamycin treatment, we see increased CCSER1, which has been tracked to a naive B cell population. TACO1, which is important for cytochrome C oxidase, and we know that mice that lack this enzyme have age-related phenotypes. And lastly, reduction of GPR155 lycose, which regulates TORC1 signaling and can potentially drive aging. So we were interested in looking at applications of aging signatures to these cells, and Nick Sham, who had led the Rejuvenome project, was part of this large group effort to do a omics sequencing of different tissues with aging in mice. And so what they had developed in the tabula muricinis was a aging signature, and their splenic signature relied upon certain genes here being upregulated with aging and others being downregulated. And so if you look at the genes that are up, we can see there's an increase from blue to green, which is the increase from young to naturally aged. And if you go back, you can see that rapamycin treated old mice sort of fall within the middle. And a very similar converse effect occurs in the group of genes within the aging signature that fall down. And so what we did, of course, what everybody usually does when you do a large omics is you build your own aging signature. And one reason to do this is the tabula muricinis is a large cross-sectional study, but it has sort of a smattering of mice of different ages and different sexes at different times, and not largely powered. Whereas we particularly look at young and at old, and we have larger numbers, as well as we cover both sexes. And of course, we found a very similar effect with genes that go up and rapamycin falls in the middle. And with those that go down with age, rapamycin sort of boost it back up, not to the level of young, but it's getting there. And Of course, there's a number of age-specific signature sets. A lot of them have been designed to identify senescent cells. So there are SenPi, SenSig, and SenMayo. And if you compare these to our custom model or our aging signature set that was derived from the tabula muris, you can see that these different signature sets are really good at identifying aging in particular immune cell subtypes. The senescent signatures are quite adept at finding senescence in fibroblasts. However, if you want to look at plasma cells, the tabula muris seems to work quite well. If you want to look at NK cells, you probably want to use either ours or the tabula muris, and that the senescent signature sets probably don't capture those. So it's really interesting to see. But really kind of drilling down into the data, what we see is that rapamycin broadly alters immune cell abundance in these aged mice. And so there's immune cell types where we see a large amount of these immune cells with youth that are then lost with age. And so this is a heat map where red is hot, red means more cells, blue means cold or less cells. And rapamycin restores them. Conversely, we see certain immune cell types build up with age, but rapamycin reduces those. And so one of these here is plasma cells. And to sort of further validate this, we do myeloanalysis, which performs differential abundance testing. And so what we can see is red are cells that are built up in control mice, and blue are cells that are enhanced in the older mice that are treated with rapamycin. And of course, we see that abundance of plasma cells. And so why should we care about plasma cells? Well, going back to Nick Sham and everybody else's work with the tabula muris sinis, is they showed that plasma cells accumulate in tissues with aging. And so you want to ask, well, are these long-lived plasma cells, are they beneficial? Are they detrimental? And our hypothesis is that they are probably actually detrimental. And so there's some recent studies that have come out that sort of fit this model. And so work done by Li Qiang and including NUS's own Brian Kennedy has showed with age that there's a buildup of immunoglobulins, which are produced by plasma cells, and that these immunoglobulins can drive senescence and metabolic decline, increasing adipose buildup, inflammation, fibrosis, and this can drive decreases in health span and lifespan. And if you block immunoglobulin recycling, you can restore some of these positive effects. Another group, Guanghui Liu, did a large omics analysis throughout the tissues of mice using StereoSeq and looking for these senescence-sensitive spots. And what they found nearby were plasma cells, high levels of immunoglobulins, and that he considers these to be potentially a part of the SAS, immunoglobulin, or the ISP, the immunoglobulin-associated senescence phenotype. And so that maybe these abundant levels of immunoglobulins can actually be driving aging. And so we have a collaboration with Derek Huffman and David Fuchsman at Albert Einstein. Derek is an accomplished geroscientist, and David is a renowned plasma cell biologist, to sort of look at whether plasma cells are truly drivers of aging. Do plasma cells become senescent themselves? Do senescent cells recruit in plasma cells through chemotaxis? And so we're really interested going forward with this collaboration. But overall, going from omics data, that we still need to confirm this and to really understand, is rapamycin just changing immune cell numbers, or is it also changing functionality? And so some of what we've wanted to do is build some better tools to do this. And so Ritu and Alina in the lab have been working on developing spectral flow cytometric panels to broadly measure immune cell lineages so that we can see different immune cell types and start to apply some functional markers. So PD-1 for exhaustion, we've added some more, but also some biomarkers of aging into there. So P16, P21, gamma H2AX, we can use MitoTracker or JC1 to look at mitochondrial dysfunction. And because we're using spectral flow, we can have 40 or more colors and do spectral unmixing, which is a huge advancement over what you can do with conventional flow cytometry, where you can maybe max out at 14 to 20 colors. And so what we've done is sort of optimize these panels for aging in mice, and just looking at these panels, what we can see in just a preliminary analysis of young versus old mice. So these are 6-month and 22-month-old mice. We can see a decrease in B cells that's very subtle, decreases in CD4, CD8, a slight increase in Tregs, changes, very subtle changes in monocytes. So but now we're looking at mice that are treated with rapamycin to see these differences. And we're doing 2 different doses of rapamycin, 42 parts per million and 126 parts per million, to better understand, is there a dose-dependent effect that's there too? But going outside of just immunophenotyping, we really want to understand if rapamycin benefits these cell types. Do they do a better job at their intended immune cell function? So for B cells, do we see better antibody production? Do we see increased BCR, so B cell receptor diversity that can be lost with age, similar with T cells? Do we see increased stimulation? How does cytotoxicity change? Importantly for NK cells, because they are there for the surveillance of senescent cells, does cytotoxicity increase? With macrophages, how do they phagocytize antigens or perform efferocytosis, which is a really important function where macrophages can clear out apoptotic cells. And it's actually an anti-inflammatory effect by doing this. And so to wrap up this part of the talk, rapamycin is able to attenuate these the immune dysfunction that we see in this mouse model of immunosenescence, where we delete ERCC1 in hematopoietic cells and drive DNA damage, which in turn drives premature immune aging. Late life intervention with rapamycin causes broad immune cell changes, and rapamycin treatment appears to be reducing the age-related accumulation of plasma cells. But moving forward, I'm in the Columbia Center for Translational Immunology. And so it's historically a bone marrow transplant unit, and rapamycin, or the orally available version of it called sirolimus, is typically used in transplant patients to prevent graft versus host disease or graft rejection. And so working with 2 oncologists at the University of Minnesota, Najla El-Jardi and Shernan Holton, we asked the question, does sirolimus, even though it's being used at higher doses to prevent GVHD in people that are receiving hematopoietic cell transplantation, could it provide some gerotherapeutic benefit while it's also preventing graft versus host disease? And this is really important because for hematopoietic cell transplant patients, they receive a pretty rough conditioning regimen of chemotherapeutics as well as whole body irradiation. And we know that iatrogenic genotoxicity can drive biological aging as well too. And so the study design was to take patients that undergo conditioning regimen and then receive umbilical cord blood transfusion, and were either on sirolimus, so rapamycin, or cyclosporin, another GVHD prophylactic, for a high dose that then sort of tapers off at day 100. Or day 100 to day 180. And typically they're given a very high bolus to cause immune suppression. And from there, they're given lower sustained doses to maintain that trough. And so our endpoints that we were looking at was the expression of P16, that classical cellular senescence marker, in peripheral blood mononuclear cells, as well as circulating inflammation from the serum. And so why would you even look at P16 and PBMCs? Well, Ned Sharpless's group about 15 years ago showed that this is an excellent surrogate biomarker for biological age, where we can see that it increases with different decades of life. We can see different biological conditions. And I've worked with other groups at the Mayo Clinic, and we've seen that exercise interventions can meaningfully reduce P16 and peripheral blood mononuclear cells, showing that even exercise can reduce senescent cell burden in older individuals. We even have preliminary data showing that P16 expression in liver transplant patients at baseline correlates with their not only their baseline frailty, but their length of stay in the hospital. And so this is what we wanted to look at. And in these subjects, we had PBMCs at baseline, day 100, and day 365. And so in black are the cyclosporin, the control group, and in red are sirolimus, or the rapamycin treated group. And we really see no difference in P16 expression. And if you look at circulating inflammatory factors in the serum, there's a number of these. One of them that's commonly looked at is GDF15, which is a good biomarker for biological aging and potentially risk of death in healthy individuals. We see marginal changes that occur, or changes that don't really have a clear trend there. And so our conclusion is that at the high doses and daily administration of sirolimus used for GVHD is less likely to confer some biological benefit to them. And this dose is very likely outside of the therapeutic window of rapamycin treatment where we see positive effects. Our study, of course, is limited by the small size. These individuals had multiple hematological malignancies, and umbilical cord is really just a subset of the hematopoietic cell transplantation donor material that's used. It makes up a very small fraction of these transplantations, as well as we're looking at immune cells. And so there's obviously going to be an influence of donor tissue on these measurements. And ideally, what we'd like to do going forward is to get a biopsy of some tissue so that we can actually see more of the recipient signal of senescence versus the donor. And so we're doing more studies with this and more studies with other biomarkers of aging in the laboratory. And with that, I definitely want to acknowledge my lab. Here we are. I want to acknowledge Ritu and Alina, Tom, who did a lot of the human work, our collaborators at the University of Minnesota, Chris Falk, who's helping us with a lot of the rapamycin work and doing epigenetic clocks, Najla and Shernan, who have both left University of Minnesota, Nick Sham, the Buck Institute, for helping me obtain the Rejuvenation project samples and our funding. And if people are really interested in immunity and aging, I also want to plug these really great and talented scientists. And from there, I'm happy to stop and take questions.
Thank you, Matthew. Wonderful presentation. I really love that you first show the people in the lab and you're also ending with it. So beautiful team. Congratulations. I really love your partly hypothesis that immunosenescence is driving senescence organ-wide. I haven't really seen that so much before. I love it. If you're looking at organ senescence, we can see when we are using P16 positive cells in the heart, in the brain, everywhere. What proportion might be driven through immunosenescence? What's your good guess?
Ooh, that's a really good one. So I think it depends on the context of what condition you're looking at. With natural aging, I think there's a lot that's there. I think if you're looking in the context of NASH, it's going to be much more metabolically driven, right? So, and I think that's what we have to do more of, this inter-organ communication of aging, right? So that, we have to look at, say, neurodegenerative disorders. There's people here that do that are looking at, do neurodegenerative disorders like Alzheimer's disease or multiple sclerosis, do they drive immunosenescence? And yes. So I think there's not only crosstalk from the immune cells to the periphery, but the periphery back to the immune cells. And it just creates this really vicious cycle.
Yeah. And of course, I'm asking you as a clinician, where do I target a certain intervention towards, to which organ? So if it would be the immune system and it would be the immunosenescence part driving then senescence in other organ systems, I would target the immune system. And otherwise I would target, as we have shown in a couple of trials, specific organ systems. Any idea what that could look like from a gerotherapeutic perspective, your hypothesis, and how our therapeutics might change?
Yes. So I think, again, it's probably going to come down to not only do you want to target the immune system, but you might want to target that tissue of interest too. So maybe you could use rapamycin for rejuvenating the immune system, and some other compounds. Say if you're looking at muscle wasting, would you want to do NAD supplementation, some of these potentially mitophagy activating compounds, right? And so I think those are ways to sort of go about it, and just showing that other groups have shown, and this is largely mouse work, that as we get older, our hematopoietic cells have this really myeloid shift or myeloid skewing and myeloid bias. And so people are using therapeutic antibodies in mice to suppress the myeloid bias hematopoietic stem cells, or HSCs. And even that is increasing lymphopoiesis, it's increasing potentially immunosurveillance, and it's alleviating a lot of these age-related effects.
Mm-hmm.
So there's ways to do it with small molecules. There's very likely ways to do it with biologics. Corinna Amor, of course, is doing this with senolytic CAR T cells.
Yeah. So I love your work about rapamycin. We are switching to that in a second. But in most of the senescence talks, we are hearing about senolytics. So you have senescent cells and you're removing that. You are bringing a new, I think, hypothesis in here, like maybe change the immune system from the inner side and let it better be there for a better function. So how do you see in your translational work, maybe in the future, how we would use rapamycin, mTOR inhibition versus the removal and senolytics? So could you think about combinations of it?
Most definitely. So there's a study that's done by Daohong Zhu where they took older mice and treated them with the senolytic targeting the BCL2 family, and it was able to rejuvenate HSCs as well, right? So that could be a really good combinatorial therapeutic. Looking forward, I think we would like to see some of that, and we're really interested in using some of these models, these transgenic models, to deplete senescent cells as well as treat with rapamycin, right? And see, does it cause an even greater effect?
Yeah. And you showed wonderfully that the proportions of immune cells are changing while being under rapamycin. So, and you also showed that the function is likely changing. How did that work in that study with regards to dosing of rapamycin? Was it also on daily or in level in these mice? Because I also want to make the link in a second to your human study.
So for Chongqing with hematopoietic cell stimulation and influenza, it was an every other day vaccination. When I did the mice for the T cell responses and everything, it was an every other day vaccination. But for Rejuvenome, where we saw the changes, that was just daily access to chow containing rapamycin. And with Joan's work, it was the weekly administration of rapamycin. And that's what largely is being used for a lot of gerotherapeutic interventions in humans. And that's what was done in the Pearl study that was sort of used to show safety, right, with rapamycin treatment.
Yeah, so that's the weekly doses of, most of the times it's 6 milligram of rapamycin, or almost. Okay, so might that be the answer that your human study did not show? I think you expected, or the hopeful for results, that because it was on a daily basis, I saw.
So it's a daily basis. So sometimes these people are starting out with 12 milligrams and then taking 4 milligrams daily to maintain a trougher, and titering it back down. So it's quite a lot. These are also hematologic cancer patients, so there's a lot of other things going on there as well too. And so, maybe we can find a way of dose escalation studies or titration studies to find maybe a window where in hematopoietic cell transplant patients, we can maintain GVHD prophylaxis, but also confer some gerotherapeutic intervention benefit.
Yeah, fantastic. I will catch your brain, because we are starting our longest study, the rapamycin one, 6 milligram weekly, how we can really look at the immune function. Switching gears a little bit, also very exciting is the plasma cells. I think a little bit forgotten. When I was a student, I always was reminded that we do not just have T cells, but also B cells. And you showed the very nice data about the immunoglobulins as a possible SAPs driving the aging process. Do you have an idea which immunoglobulins, or do we have more knowledge around that? Could we measure that in humans?
Sure. So at least in mice, it appears to be IgG.
Ah, okay.
So, and that's one of the things we want to look at too is rapamycin. Okay, so we have the transcriptomic data that shows that it's reducing plasma cells. What about serum levels of IgG, right? Like that's one of the things I want to go pull some blood samples out and look at. And so, yeah, I think those are things that could easily be measured there. The real question, I think, is, are those antibodies, are they autoantibodies?
Mm-hmm.
Don't quite know that yet. And are those long-lived plasma cells, are they themselves also producing a lot of cytokines and SASP?
Yes. Or might these be IgGs against viruses, for example, which are persistent? So do it like CMV, for example.
Sure.
Interesting. So because we know that, of course, people who are CMV and HSV positive, they have much, much more immunosenescence. So could that be the link?
And so, yeah, CMV can sort of monopolize, I think, up to like 20% of your T cell repertoire at one point. And so I think, anytime you're doing a human study, you have to check for CMV status. And there's work showing that CMV and other of these chronic viruses can drive senescence and potentially immunosenescence as well. So it's definitely there.
Yeah, great. Before going to Kalisha, who is getting many questions, I would love to ask you one question. What would you advise to young individuals coming to the field what to do in the aging field? What is the most promising topic in the aging field?
Find something that really interests you. I think, as somebody who comes from a DNA repair background and cellular senescence, we're starting to get more into the mitochondrial biology. I think it's so strong and so pervasive. And so we can knock out DNA repair broadly across all immune cells. And those mice are there. They do develop this accelerated aging phenotype. But when we knock out mitochondrial maintenance across all immune cells, it's completely lethal. So it's so important.
Okay.
And I would suggest people look there. And, there's new therapies with mitochondrial transplantation, the mitophagy activating compounds. It's a really exciting time for the mitochondrial biology field right now.
Thanks. I think your team is very happy, smiling faces on your photographs. But let's look at Kalisha. You have many questions, I think. Over to you.
Thanks, Andrea. Hi, Matthews. Thanks so much for your presentation. So let's take a look at the questions from the audience. We have the first one from Wilbert. He actually has two questions. But for the first part of your presentation, he asks, would improving immune function by Rapa in your immune-specific repair deficient animals also result in extended lifespan, or rather boost their health, particularly as Rapa so far does not seem to extend lifespan of systemic repair deficient animals?
Yes, so we don't know. We would like to try that. And what we did was with short course rapamycin treatment, eventually, if you take them off of it, the biology of ERCC1 deficiency is inescapable. They will develop that immunosenescent phenotype. It's just going to happen later on down the road. But yeah, so it's, and I think some of what was done in the ERCC1 mice before, the whole body deficient mice, that was with lower dose of rapamycin. So I think 14 and 42 parts per million, but it would be really interesting to go back and look at 126 part per million dosing and see, could we see was there just not enough dose of rapamycin given to the progeroid mice to confer some benefit?
Mm-hmm. Great. Okay, so the next question is for your second part of the presentation. Your GVHD data very nicely matches with observations from healthy kidney donors that also showed no improvements by rapamycin treatment. Do you think the timing of the intervention here played a role?
Yes, I think timing, but also really dose, right? Dose and duration is always sort of how these things work. The dosing we're using is much more sustained and at probably higher plasma levels in these transplant patients than what would be used for gerotherapeutic purposes. And of course, the daily dosing is going to be an issue as well. So yes.
Mm-hmm. Okay, so moving on to the next question by Gandhi. So you showed that rapamycin both expands the immune cell populations and also improves the functionality of existing aged cells. What kind of markers or metrics are used to distinguish between these two mechanisms in your studies?
Oh, so one is, of course, sort of, just population numbers that we can do with basic immunophenotyping. And then there you can layer on some functional markers and flow of, say, you want to look for PD-1 levels on T cells. And then what you can then do is take those cell types and isolate them and say like NK cells that express PD-1 or don't express PD-1. There's data out there showing that the PD-1 positive NK cells have reduced cytotoxicity. So you can layer this, not only your flow data onto functional data, if you sort of further stratify those cell types out, and then, see if the immune cell types actually do their intended function really well. And so that's what we're trying to do. And we're sort of using this setup not only for rapamycin, but it's kind of a game plan to test all gerotherapeutics because they can all work in different ways, like canagliflozin, a sodium glucose transport inhibitor. It increases immune surveillance, but it does it by sort of decloaking immune cells by removing PD-L1 off of the surface of the cells.
Okay, so a lot of different functional tests you could do. Another question from Duarte: could intermittent low-dose rapamycin exert a hormetic effect on mTOR that rejuvenates the immune system? And would an age-adjusted dosing strategy starting around 55 years old be rational?
So I think there's compelling evidence there from the work of Joan Manick, where I showed with the Rapalog treatment or the sirolimus treatment in older individuals looking at vaccination, right? Efficacy. And that was followed up. And so she was also trying to look at responses to respiratory infections as well. So I think the issue there for us is we always, if you want to give an intervention, you want to make sure there's a perturbation first, right? Before you give something of, do we want to give NAD if there's not an NAD deficiency? And sort of blood tests and everything, we don't have a really great reporter system for humans for measuring mTOR.
Mm-hmm.
And so I think developing tools like that could help us to determine when we could start to intervene with mTOR, as well as we really want to target mTOR signals through two sort of components, either TORC1 or TORC2. And it really seems to be that TORC1 is driving the aging effects. And so how can we bias towards TORC1 without hitting TORC2? And there's now more of these TORC1 specific inhibitors. So I think we can also get smarter with our pharmacology.
Mm-hmm. Okay, we have another similar question about dosing from Samuel, who is a GP training in internal medicine and longevity in Switzerland. So some educated patients have already started demanding rapamycin from Samuel. Would you have a dosage to suggest on a weekly or daily basis? And he premised this by saying this is not a demand for medical advice.
Yeah, so I want to start off, I'm not a medical doctor. I have a PhD, so I'm a doctor like Dr. Pepper is a doctor. And yeah, so I think the data for gerotherapeutic interventions, it's there with what's been done and what's been shown for safety in the Pearl study is a weekly dosing. And so I think hopefully as we get more of these studies, it's going to inform not only the dose, but the dosing frequency and how do we build a successful dosing regimen. But I have no clue. I can tell you how to give it to a mouse.
It's useful for the mice.
Mhm.
Okay, so we have a lot of questions on, in general, maybe policy of use of rapamycin. Do you think that rapamycin or rapalogs will ever be prescribed as anti-aging therapy, or will ethical and regulatory barriers keep it limited to disease treatment?
Yeah. So again, I think once again, for us in the US, the FDA doesn't recognize aging as an endpoint. So we have to go after these sort of surrogate conditions and everything. So if you can target it towards auto-inflammatory conditions or things like that where it may improve, that's sort of a backdoor into using it for other effects, right? I think from a payer policy, sirolimus is pretty cheap. Like it's not, going to be that expensive to give. What people may do with these TORC1 specific inhibitors, a lot of them were developed by Novartis, is you've now created new intellectual property. And okay, so for them, it's a revenue stream, but for the subjects or patients, it may actually be a better therapeutic than say sirolimus or everolimus. So, um, the field is growing, but I think we can't discount the regulatory hurdles.
Yeah, so annoying. Another question about rapamycin. It's an immunosuppressant at higher doses. How do researchers balance the potential benefits for aging with the risk of infections?
Yeah, that's really good. And so what we've seen, at least from the mouse data, is when you're operating in the gerotherapeutic window, you still have some protection against infections, is what we saw with the influenza challenge in the older mice, right? Now, the question would have to be in randomized control trials, if you took individuals and you do induce influenza in them on those that were on RAPA versus not on RAPA, what's going on there. But there's some thought that rapamycin may potentially also induce expression of some antiviral response genes.
Okay, interesting. I also have a question just from myself. So it goes back to the whole concept that your immune system can accelerate aging if it's compromised. So do you think repeated infectious burdens by, for example, COVID-19 might also accelerate aging?
Sure. Sure. And so we're trying to look at immune resilience in the laboratory, and we're doing this using sepsis. And I know Andrea and I talked about it last time when she was here, and we're trying to look at this in humans, and does senescent cell burden correlate with the severity of your outcomes in COVID or say sepsis? And similarly, can we look at this period of immune resilience, starting with mice? Where is immune resilience conferred, or does a septic event actually cause long-term aging? We're just not looking long enough downstream to see it. And sort of trying to figure out what's the window of resilience, what's governing it. Some of this has been covered by Sunil Ahuja, who has done markers of immune resilience, and how has sort of this new concept of salutogenesis, which drives immune resilience. And salutogenesis is sort of this period of good health. It's kind of like the extended warranty on your body and affect your immune system. And over time, you can sort of lose it. And these people sort of fall into three buckets of immune resilience: those with plasticity, those that can recover, and those that just degrade with challenge. And I think we have to look more and more at that. And I think even with mice, we're looking at a really homogeneous population. And that's where things like the diversity outbred mice could be a really nice tool to look at some of this as well, to sort of mimic some of that human genetic variation.
Really cool. All right. So a lot of research to be done in the future. We only have one question left in the Q&A box. It's kind of more of a general question, though. Maybe you could quickly explain, how does rapamycin interact with mTOR pathway, and why is this pathway so central to aging and metabolism?
Oh, mTOR is kind of like, everybody says like P53 is the most important pathway. And I feel like metabolism people would argue that mTOR is that too, right? It's really important for growth. It's impacted when there's sort of nutrient deficiency. It plays a role in nutrient, amino acid sensing, growth, everything. And so mTOR actually stands for mammalian target of rapamycin. So that's sort of how it was found. And so it confers a lot of things. And it's a kinase pathway that phosphorylates a lot of downstream substrates, and activates a number of pathways, such as AKT and others. And so it's kind of this master regulator that's really upstream, that sort of sits atop everything. So it's kind of the top of the umbrella. And so when you sort of poke mTOR, you poke a lot of other things.
That's a great summary of the whole mTOR pathway, which is a lot more complicated than,
Yes.
than that.
There's careers built off of it, and we still don't know everything it does.
Yeah, thank you, Matthew. It was a wonderful presentation. Thanks, Kalisha, for really handling the questions, many questions, very well. Thank you so much. What I learned is that a reminder, the immune system is just beautiful. And we just have to keep it, letting it functioning. Thank you so much, Matthew. For all of you, the listeners, hopefully you also enjoyed this very beautiful talk about rapamycin and the immune system. I will leave you with two announcements. The first is the Healthy Longevity Intensive course for healthcare professionals, which is happening at the 23rd to the 25th of February next year, 2026, and the annual conference Unlock Healthy Longevity on the 26th and the 27th of February. Both are at NUS campus. And the conference is really focusing on interventions to lower the biological age with nutraceuticals and with repurposed drugs and new ones. We are excited that the abstract submission is now open for the conference, and there is the prize attached to that. The best prize will win 5,000 Singapore dollar. We also have a startup pitch competition. Also, that pitch competition is open. Also here, there is a prize attached to that during the conference. The next webinar will be at the 18th of September. It will be done by Associate Professor Lingjang Chi, and she is looking at the visualization of metabolism in aging. I think that will be very, very exciting. We are closing the webinar with an introduction of Diffie. Take care.
We can imagine that the female reproductive system is a garden. So imagine in the garden you have the soil, which is the endometrium, and of course the seed as the embryo.